Hey you guys, welcome to 8.4 and 8.5. We're talking about enzyme activity. And so let's remember that enzymes are important phosphorylated intermediates. And so that's just a fancy way of saying that they are the middlemen between a coupled process that needs a phosphate group. So here we have one process that's exergonic, that is taking the energy or that is releasing energy to create ATP. And so ATP is formed, and let's pretend that this water is actually an enzyme. So ATP and an enzyme are going to work together to fuel another process. And so this is process one, and this is process two. And so an exergonic process is going to fuel an endergonic process. And so the enzymes are an important phosphorylated intermediate. It is the enzyme here that is labeled as water, but it's actually an enzyme that is going to be the intermediate or the middleman. So to get us started, what I want to do is I want to send you in a different direction. I want you to watch this video, which you can find the URL in the description of this video, to get yourself started on the basic concept of enzymes and induced fit. This video does a really nice job of getting it started. So welcome back, and now we're going to talk about enzyme activity. Enzyme activity is the amount of energy that it takes for a reaction to go that is catalyzed by an enzyme. And so here we have an exergonic reaction and an endergonic reaction. And these, this graph gives free energy on the y-axis and time on the x. And so we know that this is exergonic because the reactants have more energy than the products. So energy is released. And so here we know that this is an endergonic product because the reactants have less energy than the products. So during this chemical reaction, energy was taken from the environment and the products then have more energy. And so I bring this graph back up because I want to show you this very famous graph about enzyme activity. So this is a graph of an exergonic reaction. We know because the reactants have more energy than the products. And so here, what we're looking at is the amount of activation energy that it takes to start a chemical reaction. And so what an enzyme simply does is it reduces the amount of activation energy that it takes to start a reaction. So if we break this down, think about a match. If you have a match, the match has everything in it ready to do a chemical reaction, but we need to increase the energy of this system in order to light the match. It's not going to just burn sitting at room temperature. So we're going to add energy in the form of heat by striking it or by using friction to light that match. And once the temperature is high enough, once there's enough heat in this system, the chemical reaction will continue and it'll give off. So here we strike the match, we add energy, and then once enough energy is added, once we reach ignition temperature, then the chemical reaction is going to proceed and it's going to give off energy, and the products are going to have less energy. The carbon dioxide and the soot is going to have less energy than the reactants. And so if it's an enzyme, let's think about enzymes that are going to be putting two substrates together, or putting substrate A and substrate B together. And so these two substrates, these two molecules are going to be bouncing around the cell without an enzyme there, and they'll eventually meet and come together and bond, even without the enzyme there. And so enzymes just speed up spontaneous reactions. They only speed up spontaneous reactions. And so if these molecules are going to bump into one another and eventually bond, then what the enzyme does is it has a spot for each of these molecules, and it has an affinity for these molecules. And so it attracts these two molecules and puts them together. So it takes less energy of these molecules bumping around, what it does is it just takes them, puts them next to each other so that bond can form more quickly. And if a substrate is going to be broken down by an enzyme, then, or be broken down or to degrade, thinking like H2O2 or hydrogen peroxide will eventually degrade or break down on its own, an enzyme will just sort of take that molecule and give it a little tug so that it'll break more quickly. And so that is the activation energy. It is going to put those molecules together and so that the reaction can happen, or give a little tug so that it can have less energy in order for the chemical reaction to proceed. And so here, this line represents, or this line right here represents the activation energy without the enzyme. So it's going to take more activation energy without the enzyme. And so it's going to take less activation energy with the enzyme. And this here is the overall energy released during the reaction, because these are the products. Just kidding. These are the reactants, and these are the products. And so this is the overall energy released during the reaction. Make enzymes function, sometimes they require more pieces. And so we call these pieces either cofactors or coenzymes. Cofactors, sometimes people use these words interchangeably, but they are not interchangeable necessarily. Cofactors are inorganic substances like zinc, iron, or copper, whereas coenzymes are organic, non-protein molecules. And so those are going to be like your vitamins. For example, vitamin C is necessary to make collagen. What it does is it's going to attach onto the substrate, and it's going to attach not in the active site. So remember that this is not in the active site. The picture shows that this is in the active site, but I want you to remember this, that the substrate is, or the cofactor, the coenzyme, is not going to bond in the active site. Is going to bond to the outside of the active site, and it's going to change the shape of the protein. So it's going to change its conformation and allow it to perform its task in order to work. And so these are cofactors or coenzymes. Remember that that they attach not in the active site; they attach outside of the active site, and they allow for the enzyme to function. Sometimes, while enzymes need cofactors or coenzymes to start their work, sometimes enzyme activity needs to be inhibited. And so there are a couple of different types of inhibition. One is competitive inhibition. An inhibitor molecule will flood the enzyme and fits into the active site. So we call this competitive inhibition. So here, if we look at the enzyme, we have an enzyme and we have the substrate. And so this substrate is going to fit right into this active site, and the enzyme will do its job. So here, what we see is that there is a competition, a competitive inhibitor. So it will fit into the active site. It's competing for the active site. And if it gets into the active site, then it blocks the enzyme activity and no reaction can occur. An example of this is penicillin. Penicillin has an inhibitor that is going to block the active site of the bacteria from making its cell walls. So if this is the protein or the enzyme inside of a bacteria that helps to make cell walls, what we do is we provide penicillin that is going to block the enzyme from working, and it's going to block the substrate from the enzyme, and it's no longer going to be able to make cell walls, and so therefore the bacteria will die. And actually, this is interesting. Penicillin resistance occurs when the bacteria produces a separate enzyme, a whole separate enzyme. So we produce the penicillin that blocks the enzyme, and so the bacteria has figured out how to create a second enzyme, something completely different, in order to make its cell walls. So evolution is cool and interesting. Another type of inhibition, a way to stop enzyme activity, is non-competitive inhibition. And this is an inhibitor molecule that's going to bind. So here's once again our enzyme, and here's once again our substrate. But this time we have an inhibitor that's going to attach not in the active site. It's going to attach to the non-active site, and it's going to change. So it's going to allosterically attach. So allosteric means that it's not in the active site. So it's going to attach somewhere outside of the active site, and it's going to change the conformation or the shape of the enzyme so that the substrate cannot fit. And so since the substrate cannot fit, no reaction is going to occur. It is not competing for the substrate, so therefore it is not competing for the active site, so therefore we call it non-competitive inhibition. Allosteric regulation. So this is different from non-competitive inhibition. So non-competitive inhibition, the regulator, or I'm sorry, the inhibitor is going to attach allosterically. And so allosteric regulation is any case when an enzyme's function is affected by the binding of a regulatory molecule at a site separate from the active site. So non-competitive inhibition is a type of allosteric regulation. However, an enzyme can be allosterically activated or inhibited. And so in this scenario, what happens is that the enzyme is constantly oscillating between an active form and an inactive form. So here is the active form. Here is the inactive form. So here you can see that the active site is open and ready to accept substrate. And here you can see that in the non-active form, the active site is closed off and it cannot perform a function. And so the molecule or this enzyme is oscillating back and forth between these two forms. And so if we have an activator molecule, it's going to bind in right here, and it's going to sort of jam it up and hold it into the active form. So this is going to be in a stabilized active form. And so here we have an inhibitor, which is going to allosterically bind to the molecule while it's in an inactive form. And so it's going to be allosterically inhibited. And so what I have for you here is another video that you can find in the description, which is a very good explanation of this process and the process of cooperativity, which we need to understand. So this is a very good video. I want you to take a moment to continue watching this, and I'll see you in a moment. Welcome back. An example, so feedback inhibition is our final concept that we're going to cover in this video. And an example of feedback inhibition is found in glycolysis. I'm taking a different example from the book because we're going to be moving towards photosynthesis pretty soon. And so, I'm sorry, cell respiration. And so the first part of cell respiration, it occurs outside of the mitochondria, and it is a 10-step process. So here, this would be the reactants and one set of enzymes, and so here's the reaction, and so here's the products. And so here's a second set of enzymes, or here's a second enzyme that's going to fuel the second set of reactions. Here's the third set of products. And here's the enzyme number 3, which we call phosphofructokinase, is an enzyme, and it's going to catalyze the third set of reactions. And so here it's represented by the number 3. And it is allosterically inhibited by ATP. And it is allosterically activated by ADP. So remember, the end of cell respiration is going to produce ATP. And so if there is plenty of ATP being produced at the end of the line of cell respiration, if the cell has plenty of ATP, it's going to be floating around, and it's going to allosterically inhibit phosphofructokinase from working and therefore producing more ATP. This is a negative feedback loop. When there's plenty of ATP, it's going to signal the the phosphofructokinase to stop working, to stop producing ATP. But when there's lots of ADP moving around, floating around the cell, that means that there needs to be more ATP, and so it's going to bond, allosterically bond to the phosphofructokinase, and it's going to it's going to speed up the reaction. It's going to stabilize it in that on position. And so this is a feedback inhibition, when the products of a cellular pathway are going to either inhibit or are going to inhibit the production of the end result of the pathway. So I hope this helps, and I hope you have a great day.